this video we are going to look at some major questions related to CSIR UGC Life Science Paper 2. Okay, so in the top we could see questions, and in the bottom side you could see their answers. So I'm using the split screen function. That's not a big deal, but I'm kind of excited. Okay, so let's start. First question is about amino acids and peptides, all the sections. Okay, up to 49, I guess. Till that, we are going to talk about amino acids and proteins. So, which of the following statements about standard amino acids are correct? Lysine has butyl ammonium side chain. Average mass of an amino acid residue is about 110 Dalton. Universal genetic code specifies only the 22 standard amino acids. Proline has a secondary amino group. And the correct answer is C. C in the sense P, Q, R and S are correct. P, Q, R is correct. Okay. So P, Q, R is correct. Lysine has butyl ammonium side chain. Average mass of an amino acid residue is about 110 Dalton. Universal genetic code specifies only the 22 standard amino acids. Proline has a secondary amino group. Then next question, which of the following statements is not true? The pI is the pH value at which the protein has overall charge of +1. At a pH value equal to pi, a protein will not move in the electric field. An acidic protein will have a pi less than 7. A basic protein will have a pi greater than 7. The correct answer is A. The pi. That means all other options are correct. The pi is the pH value at which a protein has overall charge of +1. That is not true. All other B, C, D are correct. At a pH value equal to pi, a protein will not move in the electric field. An acidic protein will have a pi less than 7. A basic protein will have a pi greater than 7. Okay. Then which of the following statements is not true about the amino acids? Only 22 amino acids are used in protein synthesis. All amino acids exist in two stereoisomeric forms. Amino acids have an N-terminus, C-terminus and R groups. There are more than 300 different kinds of amino acids present in the cell. Not true. Okay. Third one answer is B. All amino acids exist in two stereoisomeric form. That's not true, but all other statements are true. Okay. Only 22 amino acids are used in protein synthesis. Amino acids have N-terminus, C-terminus and R groups. There are more than 300 different kinds of amino acids present in the cell. Then choose the incorrect statement about standard amino acids. Aspartate has the smallest pi value, as arginine has the largest pi value. pKa value of side chain is maximum for aspartate. Then side chain of isoleucine has maximum hydrophy value. Which is the incorrect statement? C. pK value of side chain is maximum for aspartate. But other statements are correct. Aspartate has the smallest pi value. Arginine has the largest pi value. Side chain of isoleucine has maximum hydropathy value. Okay. Then which of the following is not true about alanine? At pH 1, the overall charge is +1. At pH 1, it will move towards cathode. An equimolar mixture of D and L-alanine does not rotate the plane polarized light. It contains branched side chains, which is the correct answer D. It contains branched side chain. That is not true about alanine, but all other options are correct. At pH 1, the overall charge is +1. At pH 1, it will move towards cathode. An equimolar mixture of D and L-alanine does not rotate the plane polarized light. Okay. The next, which, I'm sorry, a bit distracting. Okay. Which one of the following amino acid, if added to an acidic solution, will have buffer reaction on solution at the physiological pH? Which of the following amino acid, if added to an acidic solution, will have buffer action on solution at the physiological pH? Which one? C. Histidine. Okay. Histidine, if added to an acidic solution, will have buffer action on solution at the physiological pH. And what is the net charge of the amino acid glutamine and arginine at pH 10.76? Negative and 0. Which of the following is an amino acid? Histidine, cysteine, glycine, proline. It is amino acid. D. That means proline is an amino acid. Remember, proline is an amino acid. And which pair of amino acids will have the highest absorbance at 280 nm? Assume equimolar concentrations. Which one? Tryptophan and tyrosine. Then, which of the following amino acid has a nonpolar and aliphatic R group? Leucine. Nonpolar and aliphatic R group. Then which amino acid residue is most likely to be found in the interior of a water-soluble globular protein? Answer C. Valine. The nonpolar amino acid residues are found mostly in the core of proteins. Okay. Nonpolar amino acid residues are found mostly in the core of proteins. The mean molecular weight of an amino acid in a typical globular protein is 110. 110. Then if the molecular mass of an amino acid is 150 Daltons, the molecular mass of a tripeptide will be 414. 414. And the closest estimate of the number of amino acid residues in a protein with a molecular mass of 85 kilodalton is 710. Okay. And if one arginine has a molecular mass of 175 daltons, then what would be the molecular mass in daltons of a circular polymer of 38 arginines? Answer is actually the B. 5928. The molecular formula for glycine is C2H5O2N. What would be the molecular formula for linear oligomer made by linking 10 glycine molecules together by condensation synthesis? C20, H32, O11, and 10. Then a protein X was fused with GFP in a vector and expressed in E. coli. The length of the protein X is 1000 amino acid residues and the molecular weight of GFP is 27 kilodaltons. What is the total approximate molecular mass of the fusion protein in daltons? 18. Answer is actually A. A in the sense 137000. So an amino acid has 3 ionizable groups with pKa's of 2.0, 10.5 and 3.8. What is the pi of this amino acid? 2.9. At the isoelectric point of an enzyme, which is 5, it was observed that there are 10 positively charged groups and 10 negatively charged groups. When the enzyme was titrated with alkali starting from pH 5 to give pH 7, 3 positively charged groups were deprotonated. The net charge in the protein at pH 7 would be B, minus 3. And imagine you are generating a site-directed mutant of a protein in which a given residue is replaced by another residue. Which one of the following residues will result in the highest isoelectric point of the mutant compared to the wild type? X to Y means residue X in the wild type is replaced by residue Y in the mutant. Which one is the answer? A. In the sense ASP to ARG, aspartate to arginine. Which one of the following statement is false? Sickle cell HP and normal HP have same value of negative charge. No, that's wrong. but all other statements are correct. Amino acids tend to be least soluble in water at their isoelectric point. That's correct. Isoelectric precipitation, remember that. Okay. Amino acids are made visible on the chromatogram by treatment with in hydrogen. Yeah, that's a practical application. So you might have done this experiment, so you know. Then the net charge on an amino acid. I'm sorry. The net charge on an amino acid is a function of the pH of the solution. That's right. Then which one of the following peptides lacks a chiral center at the first position, is devoid of hydrogen in the backbone amide at the second position, and the side chain of the third residue has a pK close to the physiological pH? 23 answer is found to be D. D in the sense given like this. Okay. Then what is the value of net charge in peptide GWYQR at pH 10.5? -1. From the following sets, pick the one that contains exclusively hydrophobic amino acids. Extremely hydrophobic. Which are they? Alanine, leucine, phenol, valine. Okay. Which one of the sequence below would be most likely termed homologous to the following? D. Then which one of the peptides can be easily detected by absorbance at 280 nanometer? 27. Correct answer is actually A. Of the groups of amino acids indicated below, which one group of amino acid can be phosphorylated? Which one? B. Tyrosine. In the cell-free lysates of E. coli, radioactive isotope can be used to label the following. Which one? B. Protein containing serine, threonine or tyrosine. And choose the mismatch pair. D. Cysteine imino group. All other answers are correct. So tyrosine group present in side chain, arginine guanidino group, histidine imidazole group. Tyrosine phenolic group, arginine guanidino group, histidine imidazole group. And presence of specific functional group in an amino acid gives a specific color reaction and use as test for amino acid. Choose the correct combinations. D. D is the correct one. Million arginine. All others are not correct. Okay. Million arginine. And the high solubility of amino acid in water is due to what? Dipolar ion structure. Dipolar ion structure. Not because of presence of side chain, not because of unipolarity, not because of the hydrophobic nature of the amino group, just because it's unipolarity. Okay. Then amino acids found in proteins that are formed by post-translational modification of one of the common amino acids, derived amino acids, include which one of the following? Which one? D. 4-hydroxyproline. Then next, choose the correct statement. 34. C. Selenocysteine is derived from cysteine. Yeah, that's right. Histidine contains 3 ionizable groups. That's not true. Among standard amino acids, tryptophan has highest molecular weight. That's also not true. And no, I'm sorry, that's true. That was a mistake. So learn from my mistake while you answer questions. And say Janet, just avoid this mistake of mistaking between correct and incorrect statements. So here we need to choose the incorrect statement, and that was turned out to be the third one. Selenocysteine is derived from cysteine. That's not correct. But at isoelectric point, protein has minimum solubility. That's correct. Among standard amino acids, tryptophan has highest molecular weight. That's also correct. And histidine contains 3 ionizable group. Okay. Selenocysteine is not derived from cysteine. Then 3 properties of an amino acid is given. Mark one amino acid which has all the three properties. Contains an amino group with an anhydride formed a yellow derivative. Is a non-essential amino acid. Most probably proline, because no, not proline. Yeah, proline. Proline was correct because I remember spraying an anhydride and getting a yellow color while performing chromatography with proline amino acid. So that's correct. Proline contains an amino group with an anhydride formed a yellow derivative. Is a non-essential amino acid. Okay, so that's all about today's question answer session. Tomorrow we'll be doing the same with next questions, 50 minutes. Thank you for listening.